Hi, I'm Doc Miller, and today what I wanted to do is sort of a think aloud of how we go from a large encompassing standard, which you might see in like a core standard or in your state standards, and how you break that down into individual learning objectives. So parsing it down into measurable and manageable pieces that your students can accomplish in one lesson, and how you can build lessons around that. This is really influenced by Doug Lemob's 4 M's from the book Teach Like a Champion. And I'm just going to kind of go through with you my thought process on kind of how I try to accomplish some of these things. You see the core standard I'm starting with, and this is from the Indiana Core Standards in Biology, about structure and function and the molecular basis for heredity. And what you probably want to do is read that very quickly, describe the basic structure of DNA, its role as the molecule of inheritance, and how it directs the production of proteins. Describe how every cell controls its structure and function by controlling the expression of its genes. Explain that proteins determine the characteristics of individuals by determining the characteristics of cells and by controlling the detailed processes of embryonic development. And you can see that the state has even listed some indicators kind of below that. The first thing that probably pops into your head when you see that is that's a lot. As a matter of fact, that would be far more information than you could teach in a single class period. And so the first step you'd want to do is identify the main ideas or the essential concepts that are coded in that particular standard into manageable or digestible chunks. And when I say manageable, I mean, could you get students to mastery in a single lesson, or at least could you get them to progress in a single lesson toward mastery? So as we do this, it's important to keep in mind that the lessons are going to have to be delivered to students based on their prerequisite knowledge. So in doing this as the teacher, you would know what they've been taught already, and you would kind of know where they are along those lines. So here's what I thought about that standard. The first thing I thought was, well, it clearly indicates that we need to know something about DNA structure and function. Okay, that's an essential part. We also need to know, because it asked about cells, how DNA replicates. All right. Now, DNA structure is a massive undertaking in and of itself, but then understanding how we get more DNA would require them to understand this, right? We also then go to something like the structure and function of RNA, because we're going to be talking about proteins and how we get proteins, and of course RNA is essential there. So we'll have to talk about the difference between the two, DNA and RNA, and then ultimately that takes us, I can't count, 3, 4, that takes us to protein synthesis, right? So really what I've done here is taken that huge standard, and I've broken it down into 4. And we would probably go even further with that one, because we would say gene expression. Well, that's going to be in genetics, right? But for my purposes of explaining to you where my mind is, this is how I broke these things down. Now, each of these 4 parts can be practiced. They can be assessed in a single class period. And then we can build activities around these ideas to further students' knowledge and to ultimately assess where they're at. You can also notice that these are the most important things there. I mean, we could very easily get lost in the minutia of purines and pyrimidines and phosphate groups and different kinds of sugars. But you ask yourself, is that the most important thing I want my students to take away? And is the academic standard really pushing us in that direction? And I think you'd find that standard, the answer is no. What we really want students to be able to do is talk about DNA and how DNA gets from cell to cell, and how that DNA goes from being information to being proteins and structures and why we do what we do, right? So once we've done that, what we've really done is made first the big ideas, the big learning objectives, right? After that, I can then figure out what I want to do to get my students where I want them to go. It's really, really hard and kind of ineffective to try to start with, here's this activity I want my kids to do, boom, oh, here's what it teaches. That doesn't really work. What we want to do is set these things first, these learning objectives clear. They need to be the most important things and they need to be measurable, and we'll talk about that here in just a second. Once I have these, I can then develop lesson plans based on the idea of what do I as the teacher need to do? What information do I need to provide? What do I need to prepare? What do I want the students to be able to do with me? And then ultimately, what do I want them to do on their own? And Doug Lemov calls this the I, we, you approach. And I think that's important. He also talks about it in something that he calls double planning. And basically what that is to create a lesson plan based on here's what you would do as the teacher, and here's what the students will be doing as their role as students. So next, we want to figure out how we're going to measure that. Now, if I were to take my initial learning objective, which I'll boil down, say for the protein synthesis portion of this, to be students will be able to determine the protein that would be produced from a given DNA sequence or genes. This is measurable in a variety of different ways. I could give students a sequence of DNA and say, use a codon chart and tell me what protein is going to be made. I could also ask students, here is a protein, tell me what the DNA sequence was that gave rise to this protein, or what were the possible options that gave us this protein. If students are capable of doing that, I can clearly understand mastery. But here's another thing to keep in mind. If we do this, the answers that the students give can tell you where they might be missing it. For example, if students, and if you're a science teacher, you know what I'm talking about, if students start listing thymine in their RNA sequence, well, now I understand they don't understand complementary base pairs and the difference between DNA and RNA. So there I can go back and I can reteach that and go, here's where I feel like we're missing it. If students are giving you the complete wrong amino acids, well, maybe they're using tRNA instead of mRNA to decode. Well, now you have an indicator of where they're missing the boat, and you can kind of start to address that, right? But the most important thing I think that we need to keep in mind is, first off, we took this huge encompassing standard and we broke it down into manageable pieces based around the most important ideas that we wanted our students to accomplish. We started with those. We made those first, and then we figured out a way to deliver that and ultimately to measure their success. All right. Now I'm going to take this one step further and show you a little trick that I learned back in my radio and TV days. It's called hotboarding. All right. And basically what you do in hotboarding is we write down each of the things that we want to do. Okay. So if I take, for example, I want to deliver information on DNA, structure, and function. All right. I'm going to place that on one Post-it note. So basically I'm going to do one for each of those standards I talked about.